let me put it this way. I think there were kind of two accidental precursors which got me into biomedical engineering, which didn't exist when I was an undergraduate at college. The first one being my own father, Edward Clifton Merrill, who was the chief chemist of the United Rexall Drug Company. And so as a child, very often I was in his lab and looking at pills and medicines and so on. And he was doing some experiments in our house with respect to how you make gelatin capsules better. And so this early background in my childhood of dealing with something to do with pharmacology and medicine was there. The second thing was quite accidental. After I did my doctoral thesis at MIT, I went to employment as a research engineer at the Dewey and Almy Chemical Company of Cambridge, which is now part of W.R. Grace. And there I came into direct contact with what we call Newtonian, non-Newtonian fluids. They're sticky, they stiffen up, they become rigid under some conditions. And so I got involved in the field of what would be called now rheology, the flow of fluids and how that flow depends upon how fast the fluid is being pushed around. And after 3 years, pleasantly at Dewey and Almy Chemical Company, and with their blessing, I went back to because I thought I wanted to teach. I went back to MIT and became an assistant professor and continued to work on the question of flow and fluids, how they flow, and what is the difference between, for example, a toothpaste of high viscosity, a toothpaste that will drizzle out of the tube, and what do you do to stop that. This interest in rheology came to be applied to medicine with Dr. Roe Wells of the Peter Ben Brigham Hospital in Boston, who was interested in the viscosity of blood. It turns out that the viscosity of blood was supposed to have been well worked out in the past several decades. But as we got further and further into it, it turned out that blood is a complex fluid, and as it slows down, it becomes more and more viscous, more and more thick, until finally it almost sets up as, for example, I wouldn't put it as drastically, but it becomes highly viscous when it's flowing slowly. This became more and more interesting as a side issue, driven as it was by my competence in industrial rheology, of the flow of oil, of paints, adhesive solutions, and so on. So fundamentally, from this beginning in the rheology of blood, the flow properties of blood, and with the tremendous help of Stark Draper, the head of the Draper Laboratory, the instrumentation laboratory, we developed a viscometer that measured this property of blood extremely accurately. And this then got us interested in other things that the chemical engineer might have some competence in. For example, in kidney treatment of kidney failure, it's desired to remove from the blood plasma of the kidney patient certain small molecules that, if they build up, make the patient very sick. That means that one exposes blood to a sheet, which we call a membrane, so designed that the small molecules will pass through, but the bigger molecules, like the proteins, like albumin, will not pass through. This is an inherent part and very widely practiced by chemical engineers. This then led us to work on the heart-lung machine, which is the case where, while operating on the patient heart, you had the flow of blood going through a machine that puts oxygen into the blood and removes carbon dioxide from it. This involves a separate type of membrane, and we, as chemical engineers, brought our expertise to work on that, and with some considerable success. And so, as these projects came on within my experience at MIT, starting around 1960 seriously, and becoming very important in 1965, I attracted a number of potential graduate students who became very interested in the field of biomedical engineering of their own accord and wanted to work with me. And they include people like Nicholas Peppers, Michael Sefton, David Tyrrell, Michael Morgan, some very skilled people who have become leaders in biomedical engineering. One of my greatest accomplishments, I would say, is having had the opportunity to get these people excited in the possibilities of improving human life and diminishing the problems of diseases by applying engineering to medical problems. In 2014, practically every chemical engineering department in the United States is now a department in effect, if not entitled, of chemical engineering and biomedical engineering. It's become an extremely important part of the biomedical engineering field. I was also very much interested in what are called polymeric materials, macromolecules. And crudely, they could be called rubbery materials, sheet materials, plastic materials, building materials. But it's the atomic and molecular structure of these that I became more and more involved in. And that led me to get involved with the brilliant Professor and Dr. Edward Salzman, who was at the Mass General Hospital, with whom I collaborated considerably. And when he and his research team moved to the Beth Israel Hospital, I followed him. And we worked for some 25 years together in one of the most effective collaborations of my memory on how do you make materials that will not clot blood. And one of the things that we came up with finally was a pure discovery. It's hard to explain why this is so. A material called either one of two things, polyethylene glycol or polyethylene oxide. And this particular macromolecule, which is highly water soluble, can be attached to various surfaces. And after it's been attached to surfaces by one technique or another, especially ionizing radiation, if the surface is exposed to blood flow, it does not incite the blood to clot. Whereas practically every other plastic or surface that the blood sees will eventually cause a set of clotting steps to occur. And this led to a whole host of products now on the market which are protected, the surfaces are protected by polyethylene oxide, or by the same molecule is called polyethylene glycol. My association with Dr. William Harris at Mass General over a very pleasant period of some 7 years, we arrived at a solution to this problem in that by using ionizing radiation to cross-link the molecules of polyethylene, while at the same time melting them so as to avoid free radicals being trapped. We arrived at a product which is demonstrably free of subsequent oxidation and refuses to break down and form particles. So one of my other contributions to biomedical engineering has been in association with Bill Harris's group to have produced a material which can reliably be used in artificial hips as a sliding surface without wearing down and producing particles. Over the years, I had all kinds of other side experiments, one of which was, and this is an important thing for biomedical engineers to be aware of, at a time there was the issue of babies born with hyaline membrane disease, a disorder of the new formed lung membrane, which led the baby essentially to suffocate. And one of the things we got into was the use of a fog, creating a fog in which we introduced the surface active material called lecithin, dipalmitoyl lecithin. And this turned out to be useful if properly administered, and it saved a number of babies' lives by exposing them to an aerosol containing this particular chemical, which you can call it a fancy soap. But the final moral of that story is, on my sabbatical year in 1970 at Children's Hospital in Boston, at the invitation of Dr. Judah Folkman, which was a great adventure for me, Mary Ellen Avery, the chief of medicine, said, well, your lecithin aerosol is of interest, but what we really should be doing is stopping the disease medically. And indeed, it was through her research and that of others that she summoned that hyaline membrane disease in the newborn is now almost a thing of the past. By proper manipulation by the physician, it can be avoided. So a lesson that I learned over the years is biomedical engineering to make products is highly useful, and it will save lives and so on, but it doesn't mean that we should not, as human beings, going beyond simply engineering, try to think of ways to prevent the disease in the first place. So going back to the issue of, for example, the artificial kidney, it would be highly desirable if we could avoid having the kidney become diseased in the first place, and that would put the artificial kidneys totally out of business. And by the same token, with respect to artificial hips and knees, if the molecular biologists could arrive at a solution to prevent the destruction of articular cartilage, it would put that out of business. But we engineers have come into this opportunity of trying to solve this problem as it exists now, and that's been highly gratifying to me. And in my association with the various members that I know of the American Institute of Medical and Biological Engineering, we all share the same enthusiasm. If we can work on something that will give people less pain, make them more mobile, make them living happier, no matter how we do it, from which discipline, mechanical, civil, chemical, we feel pleased, we feel happy. It's a profession working in which one feels satisfied.